Dear honorable viewers, all the students of MHC in entomology, Assalamu alaikum wa rahmatullah. Welcome to the discussion on the host parasite relationship, life history, pathogenicity, and control measures of mosquito. If you already subscribed in my channel, thank you very much. If not, please subscribe now to get the notification of next video. For this, click on the subscribe button below the video, then click on the bell icon and then select the option all. Very important questions for this discussion is describe the host-parasite relationship, life history, pathogenicity, and control measures of any mosquito. The host-parasite relationship, life history, pathogenicity, and control measures of Culex is discussed below. The host-parasite relationship of Culex. The female Culex feeds on the blood of man and large animals as an exoparasite. Their bites are severe nuisance to man and animals. They are important as vector of many pathogens. The female Culex acts as a host, sheltering many pathogens to complete their life cycle and helping them to infect man and animals. It carries the pathogens of dengue and elephantiasis of man. Life history of Culex. Its life history is discussed below with the following headings: copulation and oviposition. The two sexes copulate during flight. The male gets attracted to the female by her high-pitched whine. After a blood meal, the female lays eggs 300 on the surface of the water in groups forming egg wraps. Egg white, elongated and cigar-shaped, narrow and pointed at their upper end and broad and rounded at the base. The air bubbles entangled among the upper narrower ends assist in floating. After some time, the eggs become dark gray. Larva. The eggs hatch within 2 to 3 days, and a small transparent larva measuring about 1 millimeter emerges from the lower end of each egg. All 4 larval instars are aquatic and known as regular. Its body is distinctly divided into 3 parts. Number A. Head with 1 pair of antennae, compound eyes, and prominent mouth brushes, used in feeding on organic material. Number B. Unsegmented, limbless thorax, bears 3 pairs of lateral tufts of hairs. Number C. 9-segmented abdomen, bears lateral tufts of bristles on each segment. Dorsally, upon the 8th abdominal segment is located a long respiratory siphon. The tracheal openings a pair of spiracles placed at the tip of the siphon. Four small leaf-like tracheal gills are attached to the last abdominal segment. The ninth abdominal segment also bears two tufts of bristles, the ventral and dorsal brush. In a fortnight or so, the larva molts for the third or fourth time and measures 1.25 millimeter in length. At the last moult, it changes into a different form called the pupa. Pupa. Pupae are aquatic. Unlike most insect pupae, is quite active and comma-shaped, with a distinct cephalothorax which bears a pair of respiratory trumpets and are commonly called trumler. The tegument of the cephalothorax is transparent and the eyes, legs, and other structures of the developing adult are readily visible. The tapering abdominal segments have short hairs, and terminally there is a pair oval paddle-like extensions which enable the pupae to move up and down in the water. Imago. The pupal stage lasts for a short duration, usually 2 to 3 days. After the completion of metamorphosis, the pupa is transformed into the perfect adult insect called imago. Imago emerges through a dorsal split in the pupal tegument with well-developed wings and after a few moments flies away. The complete life cycle from egg to adult takes from 1 to several days. The female lives for 1 month or more and the male for about a week. Pathogenicity. Number 1. Elephantiasis, caused by a parasitic roundworm, Wuchereria bancrofti, carried by Culex fatigans. Symptoms include enlargement of limbs, scrotum, or mammae. Number 2. Dengue fever, caused by a virus and carried by Culex fatigans. Symptoms include itching, limbs, high temperature, and minute rash. Though the principal vector of dengue virus is Aedes aegypti and Aedes albopictus, Culex is another vector of this dengue virus. Control. Number 1. Elimination of breeding places. The mosquito larvae and pupae develop in water. Therefore, elimination of breeding places are of primary importance. Swampy areas and stagnant water should be drained out. The bushes and shrubs should be cleared off. Number 2. Destruction of larvae and pupae. The larvae and pupae may be killed by covering the surfaces of water with petroleum, paraffin oil, crude oil, or kerosene oil. These substances produce a thin film of oil on the surface so that the larvae and pupae die due to suffocation. The oil also enters the spiracles, blocks the trachea, and poisons the body. Emulsion of DDT, DDD, and benzene hexachloride are effective larvicides. Number 3. Destruction of adult mosquito. Mosquitoes should be killed by fumigation with pyrethrum, sulfur, etc. Residual sprays of DDT may remain effective for several months. Number 4. Defense against mosquito bite. Houses in mosquito-infested areas should be made of mosquito-proof by screening all the doors and windows. While sleeping, mosquito nets should be used to keep away the mosquitoes. After General and Applied Entomology by K. Nair, T. N. Anandakrishna, S. V. David, page 144 to 146. Thank you very much for watching and keep watching for the next video.